The global rise in myopia is a mechanical failure of the eye. The eyeball itself begins elongating, pushing the retina backward until incoming light focuses in empty space instead of on the photoreceptors. By 2050, an estimated 4.8 billion people will have this condition. A spike of this magnitude points to an environmental deficit, specifically the modern built environment. Humans now spend 86% of their time indoors, physically separated from the protective wavelengths of outdoor sunlight. To isolate the exact light frequencies that control eye growth, researchers utilize the northern tree shrew. Unlike nocturnal mice, tree shrews are diurnal, and their optical development closely mirrors human emmetropization, the process of establishing perfect focus. Testing on these near-primates revealed a highly specific mechanism. Supplementing standard indoor lighting with a narrow band of indigo light achieves 100% efficacy in suppressing induced myopia. We are going to deconstruct the exact biological bottleneck that makes this indigo band so critical. We will define a new metric for human lighting, the myopic standard, and apply it to architectural simulation. This chart measures axial length over time. Subjects raised under standard warm white LEDs experience rapid, unmitigated eye elongation, but those exposed to the precise indigo supplement maintain normal optical development, completely halting the myopic shift. The data reclassifies the myopia epidemic. It is a solvable architectural lighting failure that we can actively engineer out of our spaces. The experimental induction forces the eye to adapt. Juvenile tree shrews are fitted with a -10 diopter spectacle lens. This places the focal plane behind the retina, an extreme stimulus that rapidly forces the eye to elongate. This chart tracks refractive error under standard warm light LEDs. The black trace shows the spectacle lens inducing severe myopia, plunging to -11.6 diopters. But when researchers supplemented the environment with indigo light, the dark blue line, the myopic shift is entirely neutralized, matching healthy control eyes. This graph measures myopia suppression across wavelengths, revealing a narrow window of success. Shifting the supplemental light slightly causes steep drop-offs. Moving into the violet band at 400 nanometers or the blue band at 468 nanometers severely diminishes the eye's ability to halt elongation. Emmetropization is not triggered by generic exposure to blue light. The retina relies on an exquisitely sensitive, narrow-band frequency response to regulate its own growth. The absolute requirement for the 419 to 446 nanometer band originates in the anatomical structure of the primate eye. The retina contains a specific non-visual opsin called OPN5. Located within retinal ganglion cells, this receptor is directly responsible for the light-dependent suppression of myopia. Normally, OPN5 optimally absorbs ultraviolet radiation, peaking around 365 nanometers. But primates possess an anatomical bottleneck. The maturing ocular lens creates a physical barrier, blocking almost all light below 400 nanometers. It traps ultraviolet photons before they reach the receptors. Because the shorter wavelengths are blocked entirely, the remaining effective absorption zone is forcibly shifted to the right, landing directly in the indigo spectrum. This published data confirms the bottleneck. Figure 3a plots the mature lens transmission curve overlapping the OPN5 absorption spectrum. The intersection creates this indigo-shaded region, the precise physiological window where light can penetrate the lens and stimulate the receptors. Biology itself dictates the required treatment frequency. The limits of primate ocular anatomy turn a biological anomaly into a strict engineering absolute. Identifying this biological bottleneck provides actionable data for architectural lighting engineers. This chart defines the mu-opic metric, mapping the exact spectral sensitivity required to suppress axial elongation. The upper quartile, highlighted in red, establishes a rigid target zone. To halt myopia, light sources must provide frequencies between 419 and 446 nanometers. Achieving this does not require high-intensity exposure. Complete 100% suppression is maintained even when the environmental supplement is dropped to a minimal dose of 50 microwatts. Because OPN5 is deeply involved in circadian photoentrainment, timing the exposure is highly effective.
Applying the indigo light for just 2 hours at dawn and 2 hours at dusk maximizes the regulatory signal. It also requires a full-spectrum environment. Indigo acts as a conditional trigger that must be combined with long-wavelength white light to function properly. The mu-opic metric gives us the mathematical boundaries to move past treating myopia symptoms with corrective lenses, allowing us to build actively prophylactic environments. We can apply the mu-opic metric directly to the built environment using the LARC multispectral lighting simulation engine. Look at this model of a student seated far from windows in a standard classroom, illuminated strictly by warm white LEDs. The visual photopic lux at the desk is excellent, at 326, but the equivalent mu-opic lux is starved, registering at merely 96. Standard indoor spaces provide ample light for reading text, but actively deprive the retina of the structural emmetropization cues it needs to stop elongating. If we supplement those exact same overhead LED fixtures with the low-level indigo protocol, the simulation data shifts. The mu-opic exposure jumps to 448 lux, closely mirroring the biological protection a child receives sitting outside in the shade of a tree. Passive environmental engineering is a scalable medical intervention. It restores biological baselines without compromising visual clarity or color rendering in the room. Implementing the solution requires correcting a pervasive public and professional misconception regarding short-wavelength light. There is currently widespread prophylactic use of blue-blocking spectacles, marketed aggressively to parents as a shield against digital screen hazards for children. This creates a biological paradox. Standard blue-blocking lenses aggressively filter out the 419 to 446 nanometer band. This is the exact mu-opic spectrum required to trigger emmetropization. This consumer hardware creates an artificial blind spot in the eye's growth regulation system, effectively muting the signal that tells the eyeball to stop elongating. The gap left behind by the blue blocker directly correlates with the required mu-opic signal. The widespread daytime use of these filters by children could theoretically be accelerating the very myopia epidemic they are sometimes assumed to prevent. Reversing a trajectory of 4.8 billion myopic individuals requires precise biological alignment. We have to abandon broad fears of short wavelength light and instead precisely engineer mu-opic environments to protect the next generation's vision.